Om Swastiastu. Hello everyone, we are from Small Group Discussion Baseball. Today we will present about our student project. And the title of our student project is The Role of Enzyme in Medicine. But first thing first, let me introduce our members. So this is the members of SGD Baseball. My name is Agung Istri Anggreni. Today I will present about the background of our student project. In our daily life, we rely on enzymes to carry out our life. Enzymes are biological catalysts, also known as biocatalysts, that mean to speed up biochemical reaction in living organisms without participating in the reaction in metabolism. Metabolism is the chemical reaction that occur in the body. Enzymes speed up a reaction by lowering the activation energy, and activation energy is the initial energy to start reaction. Enzymes aid in the completion of critical tasks for survival, including muscle growth, elimination of toxins, respiration, breaking of food particles during digestion, etc. And for the next explanation will be presented by our friend.
Hello, my name is Natania and I will explain to you the background. So, in the health industry, enzymes have advantage that can be used as biocatalysts, which then cause the need for enzyme in the world to increase. As the need for enzymes increased, it has been assessed that global market for enzyme to be billions in 2010 and expected to rise at a compound growth rate. To be specific, it is 3.9% to $7.2 billion. This study clearly demonstrates that enzymes played an important role in medical field. Enzymes have great potential in health sector, for example with enzyme therapy. This fact demonstrates the significance of enzyme in medical application. We feel there is more to discover about application of enzyme in medical field or health sector. As the result, we'd like to undertake a literature review on the role of enzyme in medicine. Next, we can see the problem identification of enzyme in medical field, and then the purpose of this research, and lastly is the significances. Thank you.
I'm Chokodre Sri Agung Dalam Skawati and my name is 21-025-11252. So, the chapter 2 is about the definition of enzyme. An enzyme is a biological catalyst which mostly protein, but it can be RNA. It has function to accelerate a specific type of chemical reaction to do a lot of different purposes in the cell. Each cell contains plentiful types of enzyme molecule with specific chemical reaction. The enzyme also can be used repeatedly. So, a German scientist named Wilhelm Kuhne, as you can see in the bottom right at the screen, he discovered the name enzyme, which comes from the Greek terms meaning within and yeast. This means a yeast that has an ability to make alcohol from carbohydrates. The idea of utilizing enzyme came about in 1916 when it was discovered that it could do catalysis in chemical reaction.
My name is Ayu Karralika Raidinjana and I will be explaining about the properties of enzyme. Enzymes are composed of polypeptide and amino acid residues as the only chemical groups, such as pancreatic ribonuclease. And the combination of catalytically active enzyme with coenzymes or metal ions will form a complete biochemically active compound known as the holoenzymes. And the protein portion of the holoenzyme that starts with the apoenzyme will be covalently or non-covalently linked by the coenzyme. And from the prosthetic groups, it is divided into two. First, coenzymes, which are considered to be co-substrates because they transform chemically during the enzyme reaction where NAD or nicotinamide adenine dinucleotide is being reduced to NAD plus hydrogen or NADH. And second, enzyme change will be brought by cofactors that with one enzyme molecule remaining and are in a cycle of a chemical shift that is completed. And here I have a table that will explain about several coenzymes and the transfer of specific atoms or functional group it is employed. There are thiamine pyrophosphate, tetrahydrofolate, pyridoxal phosphate, nicotinamide adenine dinucleotide, which I mentioned earlier, and flavin adenine dinucleotide, coenzyme A, and biocytin and coenzyme B12. And here I have a different table that will explain about enzymes and their cofactors. In this table, there are 14 enzymes in total. I think that's all I have to say, and the next explanation will be continued by my friend.
Hello, my name is Fiona Natania Santoso, and I will tell you about the classifications of enzymes. Enzymes are classified based on their catalyzed reactions, sequences, and structure that are supported by biophysical models and evolution. Enzyme name consisted by two parts. The first one is substrate, and the second one ends by suffix ase that confirms that type of catalyzed reaction. The International Union of Biochemists, or IUB, makes an ambiguous name for enzyme that call enzyme commissions, or EC, to distinguish the enzyme. EC formed into four numbers. The first number will show the class of enzyme, the second number will show the subclass of enzyme, the third number will show the subclass of enzyme, the fourth number will show the connected number by its subclass. As an example, hexokinase is called ATP dihexose 6-phosphotransferase EZ2711. But this name is complicated and long, so detached from the IUB system, we prefer to call the enzyme by the traditional names, although sometimes it's still ambiguous. To limit the ambiguities, enzymes has grouped into 6 classes. The first one is oxidoreductase. Oxidoreductase is a class of enzymes that catalyze oxidations and reductions, and also catalyze the exchange of electrons between donor and acceptor molecules. This enzyme is also used as better biocatalyst due to replacing the toxic chemicals and saving the energy of resource consumption. The second one is transferase. This class of enzyme can catalyze the transfers of moieties and transfer the chemical group from one to other molecules or from the same molecules but to the different moiety. And the third one is hydrolase. Hydrolase is used for hydrolysis reaction. This enzyme catalyzes the cleavage chemical bonds and utilizing water to break some chemical bonds. So, hydrolase can divide molecule into a smaller one.
Okay, my name is Putri and I will be continuing my friend's explanation about the classifications of enzyme. So the next one we have ligase, or also known as synthetase, is used as a catalyst involving the conservation of chemical energy and provide a couple between energy-demanding synthesis process and energy-yielding breakdown process. This enzyme is mostly active during the replications, repair, and recombinations of DNA itself. The next one we have isomerase, a class of enzymes specifically able to catalyze geometrical and structural molecules from one isomer to another. Isomers named differently according to a different ordering of the bonds and or the conductivity of the bond. Lastly, we have lyases, are a class of enzymes that catalyze the addition or removal of elements such as water, ammonia, and carbon dioxide at double bonds. Lyases catalyzed reaction break the bond or eliminate between a carbon atom and another molecules, mostly found in a cellular process, for example, the citric acid cycle, or we all know as Krebs cycle. That is all from me. Thank you.
Hi, my name is Karuniaputri. We're going to be talking about the mechanism of enzymes. Enzymes are effective catalysts that drive almost all reactions in organisms, which influenced by such factors: concentration, pH, and temperature. In this mechanism, we're going to be talking about enzyme activator. Okay, enzyme activator is a chemical compound that increases the reaction rate of enzymes, where enzyme activator has the same function as a catalyst in form as ions, small organic molecules, protein, or could be a lipid. Enzyme activators are very useful in everyday life and catalytic activities. For example, there's the extremophilic microorganisms, or the best choices in industrial biotechnology. And second is glucokinase, which is function as a gatekeeper by lowering and stimulating blood glucose. And third is the enzyme glycolytic hexokinase, which proves pathological differentiation. There's a magnesium, ions, and last is fructose 2,6-bisphosphate, which is having a role for development or of cell-to therapies are caused by glycolytic enzyme.
My name is Fricillia Wijaya and I will explain the inhibitors of enzyme. Enzymes inhibitors are substances that interfere with the action and function of enzyme. Enzymes help accelerate chemical reaction in the body and are involved in many cell functions, such as signal transduction, growth, and cell division. Enzymes inhibitors are divided into two types according to their competitive skill. First is competitive inhibitors. In the case of competitive inhibition, since there is only one active binding site, the substrate and the competitive inhibitor are preventing to binding a high concentration of substrate and the substrate binds to the active binding site. And the second is non-competitive inhibitors. In the case of non-competitive inhibition, the inhibitors are binding to an allosteric site that is separated from the active substrate binding site. In non-competitive inhibition, the inhibitor can bind its target enzyme without seeing the presence of bound substrate.
Hello, my name is Gusti, Agung, Ayu, Adinda, Fidelia, Tresna, Putri, and today I'm going to explain about the roles of enzyme. So enzyme produce chemical reactions in the body. Enzymes within our body perform important roles such as building muscle, breaking down toxins, and also breaking down food particles throughout digestion. In general, the function of enzyme is to help and speed up metabolic processes in the body. Furthermore, the other role of enzyme is to lower the activation energy. And now for the roles of enzyme in metabolism. There are two types of metabolic processes in our body, anabolism and catabolism. Catabolism is a metabolic process that breaks down complex substances into simpler substances. On the other side, anabolism is a metabolic process that forms complex substances from the simpler substances. Enzymes in our body act as a catalyst. Glycolysis is the best example of a cellular catabolic process that takes place in our cells, while one of the most important examples of anabolic metabolism is the reaction of oxygen and hydrogen to form water molecules. There are 8 types of enzymes involved in metabolic processes, including oxidase enzyme, catalase enzyme, hydrase enzyme, dehydrogenase enzyme, transphosphorylase enzyme, carboxylase enzyme, desmolase enzyme, and peroxide enzyme. I think that's all about my explanation. Thank you.
Hello, my name is Putri and I'll continue the presentations about the roles of enzymes in medicine. So, enzymes have many roles in the medical world. As a diagnostic reagent, enzymes are used as materials to look for markers of a compound. Through the use of enzymes, it is possible to identify the presence of the target compound and measure its quantity. The advantage of using enzymes as reagents are that the resulting measurements are very distinctive and more specific than chemical measurements. Can be used to measure concentrations of very few compounds and are very simple and accurate. The use of enzymes in medicine includes the use of enzymes as drugs, administration of chemical compounds to manipulate the performance of enzymes to achieve certain effects, or enzymes as treatment targets, and manipulation of protein ligand binding as targets treatment. The use of enzymes as medicine generally refers to the administration of enzymes to overcome the deficiency of enzymes that should be present in the human body to catalyze certain reactions. Okay, that's from me. Thank you.
My name is Pandey Putu Karina Chandra, and today I will presenting you the summary of the group. Enzymes are biological catalysts, also known as biocatalysts, that speed up biochemical reaction in living organisms without participating in the reaction in metabolism by lowering the activation energy. Enzyme role as catalytic protein. The combination of catalytically active enzyme with coenzyme or metal ion will form complete biochemically active compound known as holoenzyme, start with apoenzyme. However, there are also other enzymes that act tightly and permanently, known as prosthetic groups, that divided into two, which is coenzyme and cofactor. Enzyme has grouped into 6 classes, that is oxidoreductases, transferases, hydrolases, ligases, isomerases, lyses. Enzyme activator is chemical compound that increases the reaction rate of enzyme. The example of enzyme activator could be extremophilic microorganism, glucokinase, the enzyme glycolytic, hexokinase, magnesium ion, fructose 2,6-biphosphate, and to slow down the action of enzyme inhibitor are needed.
My name is Ariela Gusti. Okay, I will continue the summary about the role of enzymes in medicine on chapter 3. Enzyme inhibitors are divided into competitive inhibitor and non-competitive inhibitor. There are 8 types of enzymes involved in metabolic process, including oxidase enzymes, catalase enzymes, hydrase enzymes, dehydrogenase enzymes, transphosphorylase enzymes, carboxylase enzymes, desmolase enzymes, and peroxide enzymes. Enzymes have many roles in the medical world. As a diagnostic reagent, enzymes are used as materials to specifically look for or identify the presence or markers of a target compound and measure its quality. The use of enzymes in medicine as treatment targets, drugs, or medicine generally refers to the administration of enzymes to overcome the deficiency of enzymes. According to the duration of administration of enzymes as treatment, the enzyme deficiency state can be classified into 2 states, which are temporary and permanent. An example of an enzyme deficiency disorder is hemophilia. Enzyme as a treatment target is a therapy that uses certain compounds to modify the action of enzymes in order to prevent side effects and obtain beneficial effects that target pathogenic bacteria. I guess that's all from our group presentations about the role of enzymes in medicine. We would like to thank you and apologize if there are any mistakes in our presentation. We will close this presentation with Parama Santi, Om Santi 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 Om.